Good evening and welcome to today's session. Today's session is on carbohydrate. And to get started, let's define what carbohydrates are. So to define carbohydrates, we'd say carbohydrates are biological molecules, or to be more specific, are organic compounds whose major constituents are carbon, hydrogen, and oxygen in a ratio of roughly 1 carbon atom to 1 water molecule. They derive their name from the general formula CH2ON. You should note that there is an exception to this rule in that not all carbohydrates will follow this general formula. However, because of their general formula, they are referred to as hydrates of carbon, hence their name, carbohydrates. The basic structure unit of carbohydrates is known as a saccharide, and based on the number of units a carbohydrate is made up of, it can be classified as being a monosaccharide, a disaccharide, or a polysaccharide. These groupings contain a number of dietary important carbohydrates. These are biologically important to living organisms as a source of energy and the storage of energy and as structure units. As nutrients, they can be classified as either simple or complex carbohydrates. Simple carbohydrates, or sometimes referred to as simple sugars, are those that are easily digested and serve as rapid sources of energy, whereas complex carbohydrates are those that need more time to be digested and metabolized. When it comes to naming carbohydrates, their name contains the suffix "-os". So you have glucose, lactose, mannose, galactose. So they'll have the "-os" suffix in their name. Let's now look at the individual groupings of carbohydrates, starting with the monosaccharides. So monosaccharides are comprised of only one saccharide unit, hence the name monosaccharide. Mono, one, then saccharide, the structure unit of carbohydrates. They're referred to as simple sugars because they cannot be broken down into simpler sugars by hydrolysis. They can, however, combine with each other to form more complex types. They do this by forming glycosidic bonds, which are just covalent bonds that join the monosaccharides. So the monosaccharides will follow the general formula of carbohydrates, CH2ON, with the exception of a few such as deoxyribose, which, if you remember, is the pentose sugar in DNA. So in deoxyribose, at the second carbon, instead of having an hydroxyl group, there's a hydrogen. This is why deoxyribose is an exception to the general formula of carbohydrates. In terms of physical properties, monosaccharides are often colorless crystalline solids and are sweet tasting and dissolve in water. When it comes to the classification of monosaccharides, they can be classified by the number of carbon atoms contained in their structure. So you have trioses, which will contain 3 carbon atoms in their structure. Examples include glyceraldehyde, which is an important intermediate in carbohydrate metabolic pathways. Then you have tetroses, which contain 4 carbon atoms in their structures. An example would be erythrose, which is also an intermediate in metabolic pathways. Then you have pentoses, which will contain 5 carbon atoms in their structure. Examples include ribose, and deoxyribose, which if you remember, are major constituents of nucleotides and nucleic acids. Ribose is the pentose sugar in RNA, whereas deoxyribose is the pentose sugar in DNA. And if you remember the other thing that we mentioned about concerning deoxyribose, it's an exception to the general formula of carbohydrates. Next in the classification, we have the hexoses, which contain 6 carbon atoms in their structures. Examples of these include glucose, mannose, galactose, and fructose. Fructose, glucose, and galactose are regarded as dietary monosaccharides since they are easily absorbed by the small intestines. Glucose in particular is the most common hexose. It occurs naturally and is one of the products of photosynthesis in plants and other photosynthetic organisms. It is abundant in fruits, plant juices, and glucose is considered as a major source of energy as it serves as an important metabolic intermediate in cellular respiration to generate ATP energy. Fructose is also naturally occurring and is actually regarded as the sweetest naturally occurring carbohydrate. You will find it in honey, fruits, and sugarcane. Galactose generally does not occur in free state. It's usually a constituent of complex carbohydrates where it's bonded to other monosaccharides to form the complex carbohydrates. Apart from the hexoses, there are also the heptoses, such as pseudoheptulose. And then you can also have octoses and nanoses and so on. So these monosaccharides will form the basic structure units of larger molecules such as the disaccharides and the polysaccharides. Since they contain a single saccharide unit, they can exist as an open chain form or as a cyclic form. In aqueous solution, monosaccharides are in equilibrium between their open chain form and their cyclic form. This is what this diagram here is trying to depict. So this is an open chain form of fructose, and this is the ring form of fructose. So in an aqueous solution, the monosaccharide fructose will be in equilibrium between its open chain form and its cyclic form. These structures here depict the open chain form of glucose. This one here is the open chain of ribose, and this here is the open chain of fructose. If you look at the structures, you have a carbon, which is carbon 1 in the event of glucose. So you have this carbon, which in comparison to the other carbons is different in that whereas the other carbons are bonded to hydroxyl groups, this carbon is double bonded to an oxygen atom. So this carbon double bonded to an oxygen atom this will be known, this group here, this functional group it forms will be known as a carbonyl group. And this carbon will be known as the anomeric carbon. In fructose, this anomeric carbon is at carbon number 2. This is where you find the carbonyl group. So if this carbonyl group is bonded to a carbon atom, as shown in glucose, and the hydrogen atom, this monosaccharide is an aldehyde, and it will be known as an aldose. Whereas if this carbonyl group is bonded to a carbon atom on one side and another carbon atom on another side, this biomolecule will be known as a ketone, and the monosaccharide itself will be called the ketose. This is what is being depicted here. So you have this carbonyl group bonded to a hydrogen atom there and the carbon atom there. So this is an aldose. And then you have this carbonyl group, which is bonded to a carbon atom on either side. And this is known as, this is a ketone, and this monosaccharide is a ketose. So we can say based on the configuration of the anomeric carbon, monosaccharides can be classified as being aldoses or ketoses. Glucose is an aldose, whereas fructose shown here is a ketose. So another thing that results because of this anomeric carbon, where the carbonyl group is the monosaccharides being reduced in sugars. So what this simply means is that they are able to reduce other compounds and they themselves become oxidized. So this carbonyl group is able to reduce other compounds, and itself it gets oxidized to a carboxylic acid. So because of this carbonyl group, monosaccharides are all reducing sugars. If you look at the other carbons, you will notice that the other carbons are bonded to four different four different groups. So you have, if for example, you look at this carbon here, carbon number 2 in the glucose, to be bonded to the aldehyde group here, to be bonded to this molecule here, and it will be bonded to the hydroxyl and the hydrogen there. So that forms four different groups. So this carbon is bonded to four different groups. If you observe for the next carbon and the other one, you'll find that the same is true for all the remaining carbons, apart from the last carbon, which will be bonded to two hydrogens and then a hydroxyl group. So this one is bonded to three different types of groups. So what this simply means for these other carbons is that these are known as chirocarbons, or in other terms, asymmetric carbons. So based off the configuration of the asymmetric carbon that is furthest away from the anomeric carbon, if you look at the configuration of the hydroxyl group, if that in this structure, this open chain structure, also this structure depicting the open chain structure of glucose is known as a Fischer projection. So if you look at this Fischer projection, based on the configuration of the asymmetric carbon that is furthest away from the anomeric carbon, if the hydroxyl group is on the right, this monosaccharide will be known as a D monosaccharide. And if the hydroxyl group is on the left, this monosaccharide is an L monosaccharide. So in this case, you have the hydroxyl group on this asymmetric carbon on the right, and this monosaccharide is D-glucose. And in this structure, in this Fischer projection structure, you have that hydroxyl group on the left. So this is L-glucose. Let's now look at their cyclic or ring structures. Monosaccharides can form a 5-membered ring, also known as a furanose, or a 6-membered ring known as a pyranose. So shown in this diagram, this is a 5-membered ring. We've got 1 there, 2, 3, 4, 5. So you've got 5 atoms forming a cyclic structure. And this 5-membered ring structure is known as a furanose. So as mentioned earlier, you have monosaccharides being in equilibrium between their open chain form and their cyclic form. So this is how it forms. So there'll be a sort of condensation reaction between the asymmetric carbon furthest away from the anomeric carbon. So you have that asymmetric carbon interacting with the carbonyl group at that anomeric carbon. And what you have is this ring structure formed. So an easier way to show the way this is formed, let me draw a diagram. So after the reaction between the asymmetric carbon and the carbonyl there, you have that anomeric carbon and the asymmetric carbon furthest away from the anomeric carbon being bonded to the oxygen atom. And then what you have is that this 5-membered ring form, 1, 2, 3, 4, plus the oxygen there, forming the 5-membered ring, the furanose structure. So in order to add these constituents, the hydroxyls and the hydrogens to this furanose ring, a simple rule can be followed. You can consider the hydroxyls or the hydrogens on the right as being below the plane of the ring, and the hydroxyls and the hydrogens on the left as being above the plane of the ring. So if you count, you've got carbon number 1 there, carbon number 2 there, carbon number 3, which is this carbon here. We have the hydroxyl group on the left and the hydrogen group on the right. So the hydrogen will be below the plane of the ring. The hydroxyl will be above the plane of the ring, which is what is shown here. You have the hydroxyl above the plane of the ring and the hydrogen below the plane of the ring. On carbon number 4, you have the hydroxyl on the right and the hydrogen on the left. So the hydroxyl will be below the plane of the ring and the hydrogen will be above the plane of the ring, which is what is shown here. The hydroxyl is below the plane, the hydrogen is above the plane. On carbon number 5 here, you have the CH2OH above the plane of the ring and the hydrogen below the plane of the ring. On carbon number 1, the configuration on carbon number 1, you can either have any of the two constituents above or below. So what this simply means is that if you have the hydroxyl group below the plane of the ring, this structure will be known as an alpha monosaccharide. And if you have this hydroxyl group above the plane of the ring, this structure will be a beta monosaccharide. So if you wanted to remember what makes the ring structure alpha or beta, you can use the mnemonic ABA. Alpha below, beta above. So if you have the hydroxyl group below the plane of the ring, it will be an alpha monosaccharide. If you have this hydroxyl group on this carbon, on the anomeric carbon, if you have this hydroxyl group above the plane of the ring, it will be a beta monosaccharide. So this is how this how the monosaccharides will equilibrate in aqueous solution into their ring structures or their open chain form. So you have this open chain fructose in equilibrium with its ringed structures, which will be D, alpha D fructofuranose or beta D fructofuranose. Shown here is a six-membered ring. The six-membered ring of the monosaccharides is known as a pyranose, and used here as an example is galactose. So you have that asymmetric carbon favors from the anomeric carbon again. The hydroxyl group bonded to that asymmetric carbon will react with the carbonyl group, and that will form the ringed structure. So this is alpha D galactopyranose, alpha in that at that anomeric carbon, the hydroxyl group is below the plane of the ring. This one is beta because at that anomeric carbon, the hydroxyl group is above the plane of the ring. And then you have those constituents converting the Fisher projection into the ringed structure. These ringed structures are also known as Haworth projection. So converting the Fisher projection into the Haworth projection, you follow that through. Everything on the right will be below the plane of the ring. Everything on the left will be above the plane of the ring. So if you look at carbon number 4, galactose and glucose will have almost the same similar structure, and they'll only differ at this carbon here, carbon number 4. So whereas in glucose at carbon number 4, you have the hydroxyl group being below the plane of the ring. In galactose, that hydroxyl group is above the plane of the ring. Then the other structure, the other constituents will be the same. So in the Fisher projection, you have the hydroxyl group being on the left. This is a typo. This oxygen is not supposed to be there. So you have the hydroxyl being below the plane of the ring and the hydroxyl being above the plane of the ring. This is galactose, and they will differ with glucose at that 4th carbon. The other structures just show pretty much the same. You have D ribose there, forming beta D ribofuranose and alpha D ribofuranose. You have alpha D deoxyribose forming beta D deoxyribofuranose and alpha D deoxyribofuranose. Then there you have glucose. As stated, it will differ from galactose. If you notice, it's basically the same structure with the difference being at that carbon number 4, where you have the hydroxyl group in the Fisher projection structure being on the right, whereas in the pyranose structure at that carbon number 4, you have the hydroxyl group being below the plane of the ring. That's the difference between galactose and glucose. In this diagram here is glyceraldehyde, the 3 carbon structure, the trousers were together with the dihydroxyacetone. So dihydroxyacetone is a ketone, whereas glyceraldehyde is an aldehyde. So this is an aldose and this is a ketose. Then these two here just showing D-ribose and L-ribose. If you remember from our explanation, at that asymmetric carbon, which is furthest away from the anomeric carbon, the configuration of the hydroxyl group will determine whether the structure is a D monosaccharide or an L monosaccharide. So this brings us to the next group of carbohydrates, the disaccharides. So these contain 2 saccharide units, that is, they are made up of two monosaccharides that are covalently bonded together, forming a glycosidic bond or linkage. The reaction that forms these glycosidic bonds or linkages is a condensation or dehydration reaction, which simply means that in the formation of these bonds, there will be loss of the water molecule. Common disaccharides include sucrose, lactose, and maltose. These are commonly used as sweeteners. Sucrose is made from glucose and fructose. This is simply the common table sugar. The glycosidic linkage here is a 1,2 linkage. Lactose is made from galactose and glucose, and this is simply milk sugar. It is present in milk of mammals. The glycosidic linkage here is a beta 1,4 glycosidic linkage. Maltose is made from two glucose monosaccharides, and the glycosidic linkage here is an alpha 1,4 glycosidic linkage. So this is simply known as malt sugar, commonly used as a sweetener in beverages. Shown in this slide are the structures of the disaccharides and how they form. So you have up there, this is sucrose, and you have the alpha glucose there. Alpha in that at that anomeric carbon, you have the hydroxyl group below the plane of the ring. And then you have beta fructose. So you have this glucose molecule react through a condensation or dehydration reaction with the beta fructose to form sucrose, forming a 1,2 glycosidic linkage. So you will lose a water molecule, you will lose this hydroxyl and this hydrogen here, and this oxygen will fuse with beta fructose ring, and what you get is sucrose, the disaccharide. Shown in this next diagram here is the formation of lactose, which is formed from the monosaccharides galactose and glucose. So you have this alpha galactose, you have this beta galactose reacting with this beta glucose. So the reaction will be between carbon number 1 there and carbon number 4 of the glucose, and what you form is the beta 1,4 glycosidic bond and the disaccharide lactose. You lose a water molecule in the whole process. Shown in this third diagram is the formation of maltose, which is formed from the monosaccharides glucose, from two glucose monosaccharides. So the reaction will be between carbon number 1 there and carbon number 4. This is a alpha glucose, and this is also an alpha glucose. So you have the two react, carbon number 1 there and carbon number 4. You lose a water molecule in between, and you form a glycosidic bond here, which is an alpha 1,4 glycosidic bond, and you form the disaccharide maltose. If you add water to these disaccharides, you can get back the monomer units through the process of hydrolysis. One other thing worth noting about these disaccharides is that sucrose is a non-reducing sugar, whereas lactose and maltose are reducing sugars. So this is so because you have in sucrose, you have this 1,2 glycosidic bond forming between the anomeric carbons of the two monosaccharide units. Whereas in lactose, you have this first monosaccharide, its anomeric carbon won't be free because it's involved in the formation of the glycosidic bond. Whereas for this second monosaccharide, this anomeric carbon is free, and as a result, this can form its open chain and reduce other compounds. And so this is a reducing sugar. The same is true for maltose. It will have a free anomeric carbon, and this makes it a reducing sugar. This brings us to the polysaccharides. So polysaccharides are made up of many saccharide units, more than 10 units. So if you've got 10 and below, but above 2, so 3 to 10 saccharide units, what are known as oligosaccharides, whereas 11 or more monosaccharides are the true polysaccharides. So polysaccharides, unlike the other groups of carbohydrates, these ones do not have a sweet taste, and they function as energy storage forms in plants and animals, and they also function as structure units. Examples include starch, which acts as the energy storage form in plants, and glycogen, which is the energy storage form in animals. The other polysaccharides are cellulose and chitin, which serve as structural units. So cellulose serves as the structural unit in plants and plant cell walls. It is the most abundant naturally occurring organic compound and is non-digestible by man. That is, we cannot digest cellulose. Chitin, on the other hand, is the structural component found in exoskeletons of insects and the cell walls of fungi. In terms of abundance, chitin is second only to cellulose. So if we look at the structures of these polysaccharides, you have cellulose and chitin. These will have long straight chains, whereas starch and glycogen will be branched. So you have cellulose and chitin bundling up, whereas glycogen and starch will form branches such as the ones shown here. So these diagrams here show the formation of starch and glycogen. Starch and glycogen will have a similar structure. The monosaccharide unit in both is glucose. If you wanted to name the disaccharide unit, it would be maltose, which is what is shown in this diagram here. This is the structure of maltose. So you have two maltose units form a 1,4 linkage, forming amylose, which is the straight chain form of starch. So you have this amylose again at some point in starch. Every after about 20 glucose monomer units, you'd have the amylose chain form a 1,6 linkage with another amylose chain. And this is what results in the branching in starch. So once that 1,6 linkage is formed, what you have is now known as amylopectin, or branch starch. The same is true for glycogen, with the only difference being that in glycogen, you have the formation of the 1 to 6 linkage more frequent than in starch. In glycogen, this linkage is formed every after 10 units, whereas in starch, you have this form every after 20 units. As earlier mentioned, the structures of cellulose and chitin are also similar. The difference being that in place of hydroxyl groups, on carbon number 2, chitin will have the acetoamido group, functional group. So if you look at the diagram, in the structure of cellulose, at carbon number 2, you have these hydroxyls. So these, the structure, the basic structure unit for both cellulose and chitin is beta glucose. However, you have the structures formed in such a way that you have this first beta glucose, then the next beta glucose will be sort of flipped, and then you will form the beta 1 to 4 glycosidic linkage. And what you have is that you have these alternating CH2OH groups in the structure, and then you have at the second carbon you have the hydroxyl groups projecting outward. So you have those hydroxyl groups projecting outwards, and then you have the CH2O groups alternating. The same is true for chitin, except in the place of the hydroxyl groups, you have the acetoamido group, which is projecting outwards. So the consequence of the structure of this long structure of cellulose and chitin is that because of this projecting hydroxyl groups, these hydroxyl groups can form hydrogen bonds with other cellulose structures, and what they'll form is known as a microfibril. So you have that microfibril forming. So these shown here are cellulose, long-chain cellulose structures that have hydrogen bonded to each other. And so you have these microfibrils again forming a bigger bundle of microfibrils. And because of this arrangement in structure, this gives cellulose and chitin high tensile strength. So these are unable to dissolve, unable to dissolve in water, but however, they are permeable to water. And this is important in the case of cellulose, which is found in plant cell walls. The fact that they are permeable to water, meaning the plants will be able to utilize water for their functions. And because they are unable to dissolve in water, that water won't be able to break down the cell wall. So the structures of both are unbranched and long, and they are both insoluble in water. Now let's look at metabolism of carbohydrates. The metabolism of carbohydrates ensures a constant supply of energy to living cells. Living cells will only utilize monosaccharide units in their metabolic pathways, and as such, polysaccharides and disaccharides are broken down to their monosaccharide units to be utilized in the metabolic pathways. Glucose is the most important of the monosaccharides, and the enzymes that break down polysaccharides and disaccharides to their monosaccharide units. They are most especially down to glucose, so that it can be utilized in metabolic pathways such as glycolysis or the Krebs cycle and other metabolic pathways. So if you look at this diagram here, You have starch there, which is a polysaccharide, and starch can be broken down by the enzyme amylase into maltose. Maltose is a disaccharide. So this maltose, which is a disaccharide, can also be further broken down by the enzyme maltase into its two glucose monomer units. Then you have sucrose, which we said was made from glucose and fructose. This can be broken down by the enzyme sucrase or invertase. And this enzyme will break down sucrose to its monosaccharide units, glucose and the fructose. Then you have lactose, which can be broken down by the enzyme lactase into its monosaccharide units, galactose and glucose. Then you have glycogen, which can be broken down by the enzyme glycogen phosphorase into its glucose monosaccharide units. Then you have glucose, fructose, and galactose, which are monosaccharides, and these can be utilized by glycolytic enzymes. Glycolytic enzymes are enzymes that will participate in the metabolic pathway known as glycolysis, and they'll utilize the glucose, fructose, and galactose in order to form energy. So you have, if you notice, for most of these disaccharides and polysaccharides that have been broken down, most of them will be broken down into glucose. So glucose is the most important of the monosaccharides, and after it's been produced, this glucose will then enter metabolic pathways and eventually undergo oxidative phosphorylation to generate ATP, which is used for energy for various processes. So this will bring us to the end of our discussion. We've talked about carbohydrates, we defined them, we classified them into the three groupings: monosaccharides, disaccharides, polysaccharides. We then went on to talk about monosaccharides, and we also classified them according to the number of carbon atoms in their structure. Then we talked about the common disaccharides: sucrose, maltose, and lactose. And we also went on to talk about the polysaccharides starch and glycogen, and the polysaccharides cellulose and chitin. And then we talked about the metabolism of carbohydrates. So thank you for attending. Hope to see you in the next session.